Well, folks, let's talk about bacteria. So, there are two major categories of bacteria. This first kind, so let's start with this, I guess. Bacteria, primitive cells, right? They are prokaryotes as opposed to our complex eukaryotic cells. So, they do not have all the bells and whistles that our cells do, that plant cells do, right? But even for prokaryotes, archaeobacteria are primitive. There are three types of archaeobacteria. There are methanogens, there are acidof- thermoacidophiles, and there are extreme halophiles. So up here in the upper left-hand corner, we've got some methanogens. They are producing methane gas under this water in extremely low levels of oxygen. They consume dead organic material and produce methane, and that's a flammable gas. Here in the deep sea vents, we have thermoacidophiles. They thrive in extremely hot, extremely acidic conditions, conditions in which you wouldn't think life would exist at all. And then here we have the extreme halophiles in the lower right hand corner. They love salt. Halo is salt, extreme halophile. They're extreme salt lovers. So these are the archaeobacteria. They live in extreme conditions. They're thought to possibly be some of the first forms of life on Earth because, as you should remember, life on Earth, well, I should say, the conditions on Earth long, long ago at the dawn of life were very extreme themselves. So now we move on to the true bacteria, otherwise known as eubacteria. Eubacteria, and this is really what we're going to focus on because archaeobacteria, they don't make us sick. Eubacteria can, and they do oftentimes. So we've got some eubacteria that are either heterotrophs, which means they can't make their own food, or they are autotrophs. They can. The autotrophs could either be photosynthetic, just like plants, like blue-green algae, or they could go through some chemosynthesis, which is they're using chemicals to produce their own food. It's the heterotrophs that we have to worry about because they can't make their own food. They have to feed on something else. And sometimes that something is us. Saprophytes, those are the kind of things that are going to feed off of dead, uh, dead things, right? Organic material that once was living but now is dead. We need these things around. They are our recyclers. Without bacteria, anything that dies would just sit there. It'd get picked over, but then it would just sit there and it just wouldn't decay. That decaying process is brought to you by eubacteria. It's the parasitic eubacteria that gives us some issue. These are the things that make us sick. So here is a little example of a bacteria cell. You can see that it looks very different than a virus. Looks always kind of reminded me of like a little mouse. Looks like it's got a tail. In this case, it's got two of them. Looks kind of hairy, even though those aren't hairs. Um, but the main thing is this. Look, here deep in its center of the cell is its genetic material. It does not have a nucleus. It doesn't have membrane-bound organelles. It's just got some of the vital parts and pieces inside the cell, and that's about it. The pili that you see there, those hair-like projections, those are used to oftentimes communicate with other bacteria cells, sometimes attach. The flagella, that would be plural of flagellum, those tails, those are used to help the cell move around, literally like a little propeller. You can see here the endospore. This can be used as a survival kind of technique, almost like well, as it sounds, it's a spore, like a seed. We're going to get to that in a moment. So let's move on and let's take a look at the outside of these cells. Purple and pink, what the heck does this mean? Well, we're talking about Gram stain. Gram stain is it's a certain type of stain that can be used to help us identify and kind of categorize bacteria cells. This stain only sticks to certain types of cells, and when it does stick, it leaves the cells very dark purple in color, and so they are known as gram positive. If it doesn't stick, If that stain, you put those cells in that stain, and then you rinse the stain away, and it leaves those cells pinkish, not purple, but pink because the stain is basically washed away. Those are negative gram stain cells. Now, what's the big difference? The big difference is the way their outer membranes and cell walls are made. And those bacteria that have an outer surface that doesn't allow the stain to stick, those gram negative cells, those are the ones that are more dangerous because not only does stain not stick as well to the outside of these cells, but neither do antibiotics. And antibiotics, if they don't stick to the outside of those cells, they're not going to work very well. So we've talked about mitosis once upon a time and how our cells divide and duplicate. When they go through mitosis, they make exact replicas of themselves. It's like like a copy machine. Bacteria cells, they don't go through the same process. Do they make more of themselves? Absolutely, but they're just not that complicated. And so this whole remember the dance, right? We call mitosis the dance. Well, they don't have to go through the dance because they don't have all the bells and whistles to have to sort through and divide and all that complicated mess going on inside each cell of ours. Doesn't have to happen in bacteria cells because of this, because they're so simple. Thus, we call it binary fission. So, literally, the cell just divides in two. It copies its genetics and then goes and pops in two. This kind of weird little setup called conjugation, this is actually how these cells can talk to one another. And on the left, you see how they kind of branched and they're kind of looks like they're connected in a couple spots. Well, they are, and as they are connected, they're passing information between them. You can see it again here where the pili has extended to this other cell, and boom, once it's made that connection, it is transferring information. It is literally like data transfer. And do you know what they could be transferring? They could be transferring any sort of information, but oftentimes it's information on how to be antibiotic resistant. So let's say this guy doesn't learn, but ends up being genetically antibiotic resistant. It can actually pass that genetic information onto a cell, a bacteria cell that is not currently antibiotic resistant. And once it does pass that information on, guess what? This guy can resist antibiotics as well. This is just another look at conjugation. And you can see this would be this F plasmid is what we're talking about. This is the information that could be then passed to the other cell. And you can see how it kind of just unravels. The connection is made. It is passed, well, it's copied, it's passed through. And then you have two cells with that same information. A few minutes ago, we just talked about endospores when we looked at the diagram of the cell. The endospore is it's kind of like the last resort for a bacteria cell. If conditions go south for a bacteria cell and the cell itself isn't going to survive, it can store its information in what we call an endospore. Literally, it's kind of like a seed. It's not a seed, but we can think of it like that because it's going to be very, very resistant to all sorts of conditions. And it can just sit there. It's not going to be really, active doing anything, but it's just going to wait. And it's going to wait until conditions are right, at which point it can kind of grow. Again, think in terms of a seed, how it kind of germinates into a plant. Well, this endospore would then kind of blossom, so to speak, into a bacteria cell again. And it's just a way of it being able to manage hard times. Not all bacteria cells are harmful. Some, in fact, many, are very beneficial. We've talked about how they are responsible for decay, but they also play a huge role in nitrogen fixation. And without that, plants would be in big trouble because they convert nitrogen into ammonia, and plants need that ammonia. In fact, when you put fertilizer down on your grass, one of the key components is boosting that ammonia and that nitrogen, those nitrogen levels for your grass, your yard. Nutrient recycling, we all already talked about that. Food and medicine, geez, I mean, we could go on and on about all the different food products that use bacteria, but cheeses, Swiss cheese, it's got holes because of bacteria. Um, eh, like I said, it goes on and on, but just know that when we say bacteria, it's not always just about harmful stuff. And now finally we get into bacteria shape and clustering. So we have three different types of shapes. We have spherical, we have rod shape, and we have spiral shape. Okay. But not only that, but you can also have them by themselves. You can have them paired up. You can have them grouped like a bunch of grapes, or you can have them in long strings. Okay, so you can see um, the in the upper right hand corner these single spherical bacteria cells just sitting there, right? If they're in long strings, that's called streptococci, and then staphylococci, right? So, have you ever had strep throat? Well, you had streptococcus bacteria. You actually were naming the shape and the clustering of that type of bacteria that was making you sick without even realizing it. We don't call it streptococcus bacteria infection because that's a long name. We call it strep throat because it is this type of bacteria. In fact, if you've ever skinned your knee or had any sort of cut or whatever, and you end up getting an infection there on your skin, and someone says, "Oh, that's you have a staph infection." Well, what they're talking about there is this type of bacteria cell and clustering. Okay, staphylococci. So, anyways, just a little insight to bacteria, their shape, their clustering, what they look like, what they do, and the fact that they are vastly different from viruses. Yes, bacteria and viruses can make us sick, but that's kind of where the comparisons end because they are just such different little little guys. So, hope this was helpful, and we'll see you later.